definitely one of the reasons that I get out of bed in the morning, is because I want my research to have a real world application. We really need to look after our environment for future generations. Populations of freshwater species have actually declined by 83% in the last 40 or so years. That's only the tip of the iceberg. So there are lots and lots of species living in this river, which, you can't see them here. You have to go really looking for them, and it's very, very difficult to find them. We can't protect what we don't know about. So we need really good methods of biodiversity monitoring. The University of Hull was involved in environmental DNA work from the start within the UK. I think we were one of the first universities to really start doing any eDNA research. So eDNA, or environmental DNA, is simply DNA that we take from the environment. We could take a simple water sample from this river, and basically anything that's living within that water body releases cells into the water column. So by taking a water sample, we simply scoop that up, and then we would capture that DNA by filtering it. And then once we've got the DNA on the filter, we can then either sequence the DNA, or we can use a technique called PCR, which stands for polymerase chain reaction, which basically focuses in on a particular piece of DNA from a species that we're really interested in. One of the most important priorities is to focus on invasive non-native species. So invasive species are one of the 5 global drivers of biodiversity change, and one of the key groups that we've been trying to target with environmental DNA are the drycinid mussels. So these are zebra mussels and quagga mussels that are originally from the Pontic-Caspian regions. We work very closely with Yorkshire Water and other water companies because they're very concerned about these species causing damage to infrastructure. Zebra mussels, for example, can really clog pipes, and this can cost, for example, £500,000 a year to clear these infestations. They're also damaging our more natural environment as well, so they have quite negative ecosystem effects. For example, they can actually smother native mussels, and because they filter so much water, they can actually have really quite big impacts on the water quality itself as well. We have a project that's ongoing at the moment that's funded as part of the NERC, that's the Natural Environment Research Council Panorama Doctoral Training Programme, in collaboration with Yorkshire Water. So that's a PhD project with Sarah Peixoto, and she is trying to develop environmental DNA methods for monitoring those 2 species, the zebra and the quagga mussel. And it's not that environmental DNA is a replacement for established methods as such, it's more like it's another tool in our toolbox. So, just being armed with all these different methods means that we can create a really, really good picture of the biodiversity that's out there. Not many universities really have the facilities for carrying out eDNA research. We're very fortunate at Hull to be able to have those facilities and to be able to access them, and lots of our PhD students really benefit from having that facility. I think we've got some fantastic collaborations with stakeholders from lots and lots of different groups, the conservation sector, the public sector, and it's a real pleasure to be able to work with them.